In the Buttercup Jungle lies a vicious serial killer. Day by day, animals are disappearing, never to be seen again until one fateful day when the truth finally comes out. On what appears to be any other normal day, Perry the Porcupine is found passed out in his den. His best friend, the greatest detective of all time, Henry the Hedgehog, found him in his room with shattered glass beside him. Leading up to this, Henry has been trying to contact Perry all day, leaving constant texts and phone calls. Out of concern, he heads over to Perry's den.
Oh no, my friend is dead. How could this have happened? Who does that belong to? Hey Millie, I've just found Perry the Porcupine dead in his house. I found a piece of white hair at the scene. Gather up all the animals with white hair in the station. I'll be there soon.
Understood. See you at the police station.
Seeing the evidence, Henry has deduced that the creature who committed this crime is albinism. Albinism is a genetic mutation, which is a change in one's DNA that results in individuals having an absence of melanin production, resulting in pale skin, hair, and even eyes. Because organisms with this mutation lack pigment in their cells, they are paler in comparison to species without this mutation, who typically have brighter and more colored skin, hair, and eyes, resulting in those with this mutation to have a higher chance of sunburn and vision problems. This mutation impacts the TYR gene, leading to a complete lack of thyrosinase activity, an enzyme that is responsible for melanin formation and gives different tonalities to skin to protect from UV radiation. This protein is responsible for producing the melanin that is the pigment responsible for skin, hair, and eye color, but without it, individuals experience pale characteristics. Mutations can be caused as a result of spontaneous changes within a cell, errors when DNA is being replicated, and a variety of environmental factors, also known as mutagens, such as high energy radiation like X-rays and UV light, and chemicals that alter DNA structure or interfere with transcription. Specifically, albinism is typically caused by a nonsense substitution point mutation in which one nucleotide has been replaced by another, causing the codon to prematurely code for a stop codon rather than the specific amino acid.
Here they are, Henry. Every animal in town with white fur.
Kelly Kangaroo, please join me in the interrogation room. Where were you on February 14th?
I was at the kangaroo diner with my boyfriend, Kyle the kangaroo.
What were you doing?
We were celebrating Valentine's Day by having a peaceful dinner.
If you don't mind, could I take a DNA sample? It appears you have a silent point mutation in your DNA, meaning one nucleotide has been changed, but it doesn't affect the coding for the amino acid. It would only affect your genes if it was a missense point mutation, meaning the codon will code for a different amino acid. You can see in codon number 3, place 3, that you have an A instead of a 3, but this does not affect your genes. You are not a suspect. Samantha Squirrel, please join me in the interrogation room. Where were you on February 14th?
I was hanging out in my treehouse alone.
Do you happen to have a neighbor who can verify this?
Not a neighbor, but a pizza guy came.
If you don't mind, could I take a DNA sample?
Sure.
You do have albinism, but a different type than the suspect. While the suspect has type 1 albinism, you have type 2. They are very similar, but the mutation in your gene affects a different part of the gene. Proteins are created through a multi-step process, starting with transcription. Transcription makes a copy of DNA on an mRNA strand in the nucleus, which then is later used in translation. In this process, the protein polymerase makes a mistake in your DNA, which is rare but still happens, leading to a missense point mutation. This means a nucleotide in your DNA was changed, leading to a wrong amino acid being formed. This incorrect formation means the gene is coded for a different characteristic than intended, which is why you are overall paler than the other animals in your species. All this to say you are no longer a suspect, as your DNA does not match.
To expand on this, in transcription, which is the process of copying DNA into RNA, helicase firstly separates the DNA double helix. Next, RNA polymerase lines up RNA nucleotides to along one strand of DNA, following the base pairing rule in which A always pairs to T in DNA and U in RNA its complementary base pairs, and C always pairs with G its complementary base pair. Complementary base pairs are set ways the nitrogenous bases of DNA and RNA attach, being necessary to ensure proper formation of the nucleic acids. In the next step of transcription, the single-stranded mRNA that was produced peels away from the DNA and the DNA strand rewinds. This process has to occur in the nucleus, as that is where the DNA of a cell is stored. RNA is used in this process as it allows for genetic material to be transported out of a cell's nucleus, something that DNA is not capable of and used for protein synthesis. There are 3 different types of RNA. mRNA, also known as messenger RNA, is, as its name suggests, the messenger, coding for specific proteins. tRNA, also known as transfer RNA, is responsible for transporting amino acids to the site of translation. rRNA, also known as ribosomal RNA, is responsible for translating mRNA into protein. Overall, the process of transcription is very similar to the function of a black and white copying machine, in which when a colorized input is put in, it returns an output that is a near duplicate of the original, being in the same format and size as the original but having a different color palette.
Mr. Tiger, please join me in the interrogation room. Where were you on the day of February 14th?
I was out seeing Zootopia with a group of friends.
Can I see your ticket as proof? Okay, as one last precautionary measure, can I take a DNA sample? You do have a mutation, but it is not albinism. You have leucism, which can appear similar but results from a different mutation entirely. This mutation most likely occurred during translation. Translation is a process where mRNA is transferred to tRNA and brought to a ribosome, where it is then changed into a specific line of proteins, which codes for specific functions. In this process, RNA polymerase, a protein used to match nucleotides, made a mistake, which led to your mutation.
To expand on this, we need a few terms. First, messenger RNA or mRNA is what carries genetic information to the ribosome and then serves as a template during translation. Next, transfer RNA or tRNA brings amino acids and transfers the correct amino acid to the ribosome based on the correct mRNA codon. A itself has anticodons that pair with the mRNA codon. Lastly, ribosomal RNA or rRNA has both functional and structural components that help catalyze peptide bonds between amino acids. These components all work together in translation, which happens in the ribosome. Translation starts when the first transfer RNA binds to the codon AUG of the messenger RNA, which is also known as the start codon as it is always the first in the sequence, making a temporary hydrogen bond with the mRNA. tRNA then brings amino acids to the ribosome, which links them together in the correct order based on the mRNA sequence. It links them together with peptide bonds, a strong covalent bond that forms between 2 amino acids. This process repeats, making a polypeptide chain until the tRNA reads a codon of UAA, UAG, or UGA, which all code for stop on the mRNA, and translation ends. The protein that was formed is then released. Think of this process as breaking bread. The recipe represented the mRNA, U being the tRNA, and the kitchen the ribosome, and the amino acids as the ingredients. First, you start the recipe at the top, like how tRNA starts with a specific codon. Next, you gather ingredients based on your recipe, similar to how tRNA gets amino acids based on mRNA. Finally, you put all the ingredients in the order the recipe says, and the tRNA attaches the amino acids in the order the mRNA says, until a stop codon, or your recipe gives the signal to stop. Additionally, all of this happens in your kitchen, or the ribosome.
Fred Farrett, please join me in the interrogation room. Where were you on the night of February 14th?
Nowhere.
Fred, where were you? Frederick Farrett, where were you on the night of February 14th? It appears as though you are Perry's neighbor. Were you on good terms with him?
I don't know. I knew him, I guess.
Fred, please answer my question fully.
We weren't really close. I didn't talk to him much, though I heard a sound coming from his room that night.
That's suspicious. Okay, as one last measure, can I take a DNA sample?
Sure.
Fred's DNA is very similar to the culprit's DNA.
Okay, we should head back to the scene to gather further evidence and insight.
Henry believes that he has solved the case, deciding to have one final look to check his suspicions. However, when he goes back to the scene of the crime to check his suspicion, he finds a strand of long, white, albino hair on Perry that is undoubtedly Millie's, who had framed the case on Fred, causing a shocked Henry to discover who the true culprit is. Upon this discovery, Henry rushes back to the police station.
Millie, how could you?
Henry, I don't know what you're talking about.
How
This
didn't I
strand of
figure it
hair
out?
proves
How didn't I
nothing.
suspect it was you? Every single thing pointed to you, and I still couldn't figure it out.